What's up, man? Get up, man. Get up, nigga.
Can't bust me.
Get up, you gotta do your verse, nigga.
Alright. Yeah.
Don't be such a square. Everybody who's anybody drinks.
Yeah. Only when I'm drunk. Only when I'm drunk. Alright. Think I'm feeling a little better. Ready to bust this. Yeah. Only when I'm drunk. Only when I'm drunk. Yeah. Only when I'm drunk. Only when I'm drunk. Alright. Think I'm feeling a little better. Ready to bust this. Yeah. Only when I'm drunk. Only when I'm drunk. Yeah. Only when I'm drunk. Only when I'm drunk. Alright. Think I'm feeling a little better. Ready. Yeah. Only when I'm drunk. Only, yeah. Only when I'm.
And now,
Yeah. Only when I'm drunk. Only when I'm drunk. Alright. Think I'm feeling a little better. Ready to.
Sorry about that, fellow Cannabisians. I had a rough ass night, so sorry I started this stream too late. How you doing, Paul? Good morning, good morning. Alright, hold on. I'm trying to pump this music, and for some reason, we're not getting the love that I'm trying to get. Alright. I hope everybody's doing okay. There we go. I had to set all this shit up over again. Yeah, so I hope that's a little better. Last time I was playing this, it didn't come out too loud. So this one's all about breeding. I got a couple YouTube videos, a couple articles, and a podcast, I guess YouTube, Shaping Fire with Shango Los. And it's all covering breeding. Let me go ahead and type it in here. Let me respond. You had a rough night. Didn't sleep too well. Go ahead. I couldn't even spell Paul. That's how bad it was. So I'm gonna go ahead and get us into this article. Yeah, I'm gonna get us into this article. Bam. That worked right. Okay. Let me get out of here so we can read this article. What's up, right? No, okay, that's why. I ain't going through all that. So here we go. This is from Floriflake. Actually, this is one of the article I wanted first. Okay. Seeds here now. I did want to do this one. A guide to understanding the science behind cannabis breeding. Now, I'm not sure where everyone is as far as breeding goes, but there is a P1, a F1, a S1. P1 is the original parents of whatever the offspring is, the cross, which is the F1 generation. Now, S1 is taking basically a P1, cutting a branch off, using a reversal spray, whether colloidal silver or sodium thiosulfate, and coming up with a concoction that basically blocks the female hormone of the estrogen of the female plant to make it produce pollen. Now, it doesn't produce as much pollen as a male, maybe actually 25% of the pollen that you would get from a traditional breeding, male and female, with just a female. But once you cut that off and clone that, that is the S1, which is a little different from having two original parents crossing and making an offspring, which would be the F1. The S1 is basically the parent is also the offspring. So now that we've established that, let's get on down a little bit. All right. Let's just get down to the nitty gritty because I got a lot to cover and I don't feel like reading. I got a headache that's kind of messing me up. But my word's my word. I go live when I said I go live, even though I'm 15 minutes late. But cannabis breeding is deliberate manipulation of a plant's genetics to create new and improved cannabis strains with cannabis trait. The goal of the breeding is to enhance specific characteristics of the cannabis plant, such as flavor, potency, yield, resistance. My favorite is color. To achieve this, breeders use a variety of techniques. The process of cannabis breeding requires a deep understanding of cannabis genetics, including the role of the different genes and how they may interact to produce specific traits. Okay. So I hope that's simple enough. To kind of explain what they're talking about and also what most people are doing when they're breeding. Is let's say you have 100 seeds. 25 of those seeds are going to more represent to one of the parent traits. The other 25% will be the second parent trait. So we'll just go 25% male, 25% female for this example. The other 50% is a mixture of the two parents combined. So pheno hunting is basically trying to figure out which expression you're really looking for. Say you do sift through that 50 that represents the parental traits. How extreme each one of them is does depend on each individual plant. So you do want to pick the one that is specific for you. Okay, maybe this one say has a licorice taste. You want it to be way more pronounced than what you're getting off of him. So hence you have to keep hunting and hunting through different plants to get the exact traits that you're looking for. Excuse me. So dominant genes that are expressed traits that are paired with a recessive gene. Recessive genes that express traits only when paired with another recessive gene. So something like a pink hairs, purple leaves, purple hairs, things like that are recessive traits. So you're going to have to get two plants that have a strawberry flavor to get the end result being total strawberry. You're going to have to get two plants that hold some kind of purpling anthocyanin, and you're going to have to mix them together. But it's going to be extremely hard to get two green parent plants that don't really represent purple traits to have an offspring that does have purple traits. You have a much higher chance if you have two parents that represent purple traits. More than likely, the offspring is going to be purple. If you're going for pink hairs, you're going to want to mix two parents that represent pink hairs on each of the parents. Hopefully the offspring, both of those traits will carry over. And so codominant genes that express a mixture of traits when paired with another of the dominant genes. So this is where I guess you could get purple leaves and pink hairs when you get these two dominants getting together. Codominant. meaning depending on, I guess, the lock of the genetics, another certain plant could bring it out. Let's say Northern Lights and Strawberry Cough. When you mix them together, you get an extreme tropical flavor. If you mix the Strawberry Cough with Bubba Kush, that strawberry flavor is totally lost and say goes into a berry. So it does matter what parents you're mixing with. They will each come out with different flavors, traits, expressions. All right, so cool. We covered that. And if I ramble and miss something, I have some videos that kind of cover this. Yeah, screw it. Okay, the importance of plant breeding. I think we kind of went over that to give you a better end result than what you started with. Is ultimately the point of cannabis breeding, to get the flavors, the plant structure, the colors that you're looking for, the effects you're looking for. So I can go ahead and summarize that. Nice AI. So I don't even know if I need. Oh, Nicholas, how you doing? Good morning. Trying to get over a cold and keep breathing at the same time. I guess talk about breathing while I'm breathing a cold. But do I even need to go over traditional plant methods? I can skip it if need be. Just let me know. Okay. Traditional cannabis breeding refers to selective breeding and backcrossing. Selective breeding is a process where breeders cross plants with desirable traits and use a parent plant in the next generation. By selecting plants with desired traits, breeders can create new strains with specific characteristics. Backcrossing is a process where breeders cross a hybrid plant with one of its parent plants. This process is used to reinforce desirable traits that from the parent into the hybrid. Breeders may use this to create stable strains with consistent characteristics. So the way I understand it is going F1, F2, like breeding in those generations is going forward with the process. Back crossing is going back and locking in one or two specific traits. So what makes, I guess, breeding so complicated is you have to keep the original plant that you started working with. And the way to achieve this is just take clones. Because I do realize after time, by the time you grow the plants out, force them in the flower, dry everything out, the clones come back. It's a long process, and you've got a huge plant while you're in this holding space to even find out if you want it. So the way to kind of get rid of the mother plant, having a big bulky plant in there, is just take clones, mature the clones, and you still have the original plant in essence. That's in theory, is what I'm trying to say. It's they do say there is genetic drift, but depending on how you take care of a plant, how stable your environment is, that's something a lot of people say can be avoided. No scientific evidence, though, I do want to say that. But to be honest, there's hardly any scientific evidence on cannabis. Alright, so both selective breeding and backcrossing is time-consuming processes require a lot of patience and attention to detail. However, they are valuable methods for creating new and unique cannabis strains. And also a big key is to take notes on everything. I'm kind of loose with my notes. I noticed, I guess I'm cheating myself. I don't feel I'm as professional, so to speak. Let me kind of see the chat. My bad. I'm missing this. Okay, thank you, everyone. Everything is, I guess, decent. So, taking vigorous notes. Now, you want to almost be a nerd at this point. I recommend taking notes on every aspect of growing. Have a note every week, some kind of detail that you noticed about the plant. Once you pop the seeds, how quick is it to actually start growing the real leaves that you're looking for? Let's see what else. How the body structure was week 1, week 2. Did it have any mutation like topping itself or something like that? I see that happen quite often. But every step of the way, if you notice a color difference, hey, this stem is thicker than the rest. This plant is a more vigorous grower. This seems to have more overall health. Hey, this body structure and height is what I'm going for. You want to detail each of the plants that you're going for. I kind of start out with 10 seeds. Sometimes all of them those sprites, so I might start with 8 plants, but I'll pheno hunt through that to see what I like. Then do another round. If the original round holds up, I'll keep that and go to the 3rd round. But if the 2nd round is better than the 1st round, we'll go from that. But that you can slowly but surely pheno hunt. You don't have to feel that you have to pop 100 seeds or 1,000 seeds at once. Okay, so tissue culture. This is the one I understand that this is cutting edge technology. But basically, this is like freezing eggs to try and have babies past your prime. You need a lot of electricity, a cryo center, and I guess deep pockets to keep this thing in refrigeration. I understand trying to hold plants and hold seeds for later. You know, I get some good stuff, but really it's not going to come up until about 10, 15 years, 20 years down the road. People are looking for all types of old strains now from like the 70s, 80s. And I get it would be nice to have, but do you really want to pay that bill for 20 years for your stuff to become marketable? I don't think the cost effectiveness is going to weigh out. Your return on investment will never be met. Unless it's like $50 a month, but I'm figuring it's probably like $1,000 a month to hold your shit. So 20 years of that, yeah, I'm not a millionaire, and who knows what Cannabis is going to be doing in that meantime to get your money up to even pay that. So this is the professional way. I understand tissue culture, but for us, I don't know how feasible that really is. This is below freezer temperatures. Okay, genetic modification. A technique used to grow new plants in a small piece of plant tissue in a sterile lab environment, basically like a petri dish and growing it up. This allows breeders to produce genetically identical clones and maintain the desired traits. A technique used to modify the DNA of the cannabis plants, allowing breeders to introduce new traits or remove unwanted ones. This can be achieved through a technique such as CRISP. Okay, yeah, it is that like DNA cryo freezing crap. Marker assisted breeding. A technique used to identify and select specific cannabis genome associated with desired traits. This allows breeders to more effectively develop new strains and reduce the time and cost of breeding. This still is all like high tech and too professional for me. Okay. Modern techniques in cannabis breeding have allowed more precision and controlling and developing new strains. Sorry, I don't have my contacts in. My eyes are so puffy, I can't take it. I tried for like 10 minutes and my eyes were just burning with contacts in, so I'm sorry. I've been wearing glasses since I was 5. I'm 47. Sorry that this is so bad. But I am still the best live streamer. That's a lie. Breeder Skin and Ziva. Oh yeah, I just read that. So sorry. Yeah, best streamer ever. I knew that. Okay, understanding genotypes and phenotypes. I'll try and spare you from the reading. But the genetics of a plant, the traits, the expression you plan to see out of a plant. An Afghani versus a sativa. Afghanis are short, bushy, and sativas are long, lanky. So that's what you're going to expect out of the genotype. A phenotype is the genetics plus the environment will give you the phenotypical expression. Me growing the same exact clone that someone in California would have, me growing it in Delaware, Vermont, we're going to get different results. The terroir, the elements, and the soil type is going to play a part on the final results that we get. The temperature, wind, everything. We might have plants identical in height, identical in effect, but say if I'm dealing with more wind, my buds are going to be more oily and waxy, basically trying to combat that, versus somebody who's in full sun, they're going to get a lot more resin and THC content trying to block out the UV rays. So the phenotypical expression will be different, even though the genetic is still the same. I hope that makes it. And there's a name coming up, Gregor Mendel. He is actually the person to coin the phrase genotype and phenotype. He was an agriculturist turned bishop, I believe, around the 1850s. And we still use a lot of his techniques and theories to this day. That look good. So that would be, this is a good point for what I'm saying. This person growing this bud and the environment that it is got him this moisture content, this result, this health. Me growing it with half the life, you would understand it would be a lot less bulk on this. And that would be my phenotypical expression because the environment wasn't as conducive as this one to optimum growth. So even though we would have the same plant that has the potential to do this, if you grow it totally wrong, you're not going to get this. So there is genetic DNA code in plants that you're expecting to unlock, but without the correct environment, without the correct 10 cardinal parameters, you're not going to get the phenotypical expression that you're looking for necessarily in plants. That's the thing I wonder about the tissue culture thing, because maybe you're saving genetics from something that was grown in Spain and you trying to replicate that, growing it in Nevada, might not get you the memory that you had in your head. We'll see. So here we go. This is the best I can do because my eyes is getting tough concentrating at the screen. Strategies for successful cannabis breeding. Successful cannabis breeding requires careful planning and attention to detail. I think I said that. I was got that. Select the right parents. Controlling pollination is huge. It seems that a lot of our legendary weed strains are accidents. exactly what they're controlling, talking about. Not controlled pollinations. They either harm or were pollinated by something that did harm. I'm referring to our sour diesels. I guess not super skunk. What the fuck is that? Not Bubba Kush. Even though I think that's an accident too. Gelato is one. I think it's a harm of Sunset Sherbet and something. But you definitely want to have some kind of room or a grow tent, more preferably, and turn the fans off at this point. You do want to have a pollination session where you give the plants about 3 days to pollinate all it's going to pollinate, then turn the fans off, and that will help to control the pollination. It's not going to have as much force to travel to other plants, other portions of the room. And you know your results are exactly what they are. This is coming from this plant, breeding with this plant. And I've had mysteries before. Okay. Maintain genetic diversity. Okay. And I'm sorry, there's also an echo for me, so it's really distracting. Maintain genetic diversity. So, like I was saying, you want to pheno hunt the hell out of this. You want to pop 100 seeds, I would say, but it doesn't have to be all at once. These plants take up about 4 feet of space up and down if you're lucky. When you're hunting, they actually say you want the plants to go a little more than what you would normally grow it, just to really see what the plant will do. So give it an extra, say, 2-3 weeks of veg. Give it an extra week or two of flower, just to see how it handles senescence, growth with the 7 stages of cannabis plant life. Monitor and record everything I told you, and experiment with different breeding techniques. Now, the only thing I would really recommend, I think most people are fem only growers at this point. I'm not live. Yeah, I'm not live. The chat is telling me something's wrong. Where's the... Oh, there we go. Sorry, I forgot to keep on. My fault. Let me go to the music stuff. My fault. Let me go to YouTube and see what the fuck is up. Yeah, I ain't meaning to do that. I thought I was getting it. Yeah, I thought I was getting that. There you go. Yeah, my fault about that. So why is this? Yeah, I don't see this shit. Okay. So let me go on to YouTube. I don't know what's up with the mobile app on YouTube. YouTube Studio is what I'm referring to, actually. Yup, you not faking. You are not faking. Yeah, it's definitely not saying I'm alive. Hold on. I do gotta go to. Oh, okay. How you chatting with me and I'm not live? I'm not understanding. No. I didn't mean that. I didn't mean that. No. Okay, hold on. Can I restore this shit? I did not mean to do that. Okay, cool. So I can at least recover this part. Let me go again and get where we was at. And then I'll just get to the what? Oh yeah, I do need that. There we go. Everything back. Good. I was worried for a second. Hold on, yeah, give me one moment. I need a break real quick. All right, my bad. I'm sorry. I drank too much and got nauseous. I didn't get sick, but yeah, I had the whiz and splash some water on my face. So where were we? Where was we? I'm gonna get rid of that and bring this back up. Okay. Yeah, that scared me. I lost this page. That's the other part. I had to get this back. Yeah, I don't really care about the future of cannabis breeding. But we're going to this. Floraflex. Okay, we did that. So this is what I wanted to go into. The breeding technique. Open pollination allows cannabis plants to naturally pollinate each other without human intervention, which promotes genetic diversity. So this is basically cannabis in the wild was done for millennia. A field of it would grow, males and females, and the males would just get whatever female they got. So it's not scientific, but like they said, genetic diversity. The best crosses you get are two strains that are opposite ends of the spectrum. If one is, say, chocolate and licorice, you want one that is, I don't know, let's say earthy and fruity. When you mix all of that weirdness together, out of all the seeds you'll pop, you're going to get the most unique thing you can possibly get. Open pollination is a form of getting something like that to happen. You don't really know what it's crossing, so there's a lot of great potential, but there's also a lot of bad potential. Selective breeding. Hand pollinate specific plants to create controlled crosses and selections for desired traits and offspring. Back crossing. Crossing a hybrid offspring with one of its parents to reinforce and stabilize desired traits. So to go back over back crossing again to lock in a specific trait, they're saying do it with one of the parents. This is still based off of male and female. But if one of the parents has purple, the other one is green, and you're going for purple, of course, back cross the offspring with the more purple parent to reinforce that purple trait. Hybridization, crossing two distinct cannabis varieties to create a new and unique combination of trait. Documenting, tracking everything. So the legal considerations, this seems tricky to me. This is something that I hear cannabis breeders talk about all the time on Future Cannabis Project and Shaping Fire. We're going to use one of their podcasts today. But they talk about this. How can you really say who owns what? We're all working off the backs of someone else's work. So to try and claim something new is really iffy. This seems like some industry, like the government getting in BS that's going to screw the common person over. We're never going to get like an OG Kush again, which was Bubba Kush mixed with Indiana bubblegum. Bubba Kush originated in Florida, like the Florida Triangle. I think that's also a cutting of kryptonite. What the hell is this shit? Triangle Kush. Triangle Kush, OG Kush, and Bubba Kush kind of all have the same parents. They're just expressing different phenotypes. The environment they're growing in makes them express different things. within the genetic code. So the genotype has 100% potential to do whatever the genes have. You're trying to get a percentage of that 100% mixed with another percentage of 100% to get a new 100% from the offspring, which would be the F1s from the P1s. So I don't know if this really goes to us. I pretty much think if you came up with the cross, can prove it, legitimize yourself, come out with it. I don't really see what the government patent officer has to do with you. Are we done with that too? Nice. Okay, so I can bung that out. Oh, I never went to corner camp. That's funny. So let's see him. Oh, this video. Okay, something's up.
P, F1, and F2 crosses.
What is up with that? Alright, what is the video? Okay, yeah, that's exactly what I want. Okay.
In this video, we'll walk through the P, F1, and F2 crosses that taught Gregor Mendel the basics of trait inheritance.
I get you. I see what's going on now. So yeah, let's get into the video portion. The best live stream ever. All right, on a good note. Okay. So first off, get that queued up. Get out of here. Get back to here. There we go. Video.
In this video, we'll...
I think they start talking off the rip. Okay.
walk through.
Something's up with the... I don't know why OB's kept resetting itself. So I basically have to, it's monitor the volume and the output, or no monitor, and I have to switch between the two to reset.
Through the P, F1, and F2 crosses that taught Gregor Mendel the basics of trait inheritance.
Here we go.
In the 1800s, Gregor Mendel worked with pea plants to determine how traits are passed from one generation to another.
He worked pea plants, P1s. He took parent to do his experiment, is basically what they were saying.
One trait he focused on was pea color. He noticed that there were two variations in pea color: plants that produced green peas and plants that produced yellow peas. To learn about how pea color was inherited, he set up a series of crosses where he mated pea plants together and observed the pea color in their offspring.
So, he took plants that showed yellow and plants that showed the regular color. Now, this is what cross hybridization is. They're saying two different plants to get a desired result. So he started out with this as his original parents and just measured what the offspring, the traits that they express, off similar to what the parents were.
He started this process with two different pure breeding plants. A pure breeding plant is one that only carries one type of genetic information associated with the trait of interest. So in this case, Mendel started with a pure breeding plant with yellow peas and a pure breeding plant with green peas.
Pure breeding or heirloom basically original genetics or what we seek out of land races. Basically, it's been so crossed that it's only going to show these traits. There's going to be no deviation. That's what the P1 you're looking for. Something that's not a bunch of crosses put together, but as pure as you can really get the original parents. That way you can see how well the traits are transferring. And that's the point that Mendel is at.
These two plants are the P generation. When Mendel crossed the P generation plants together, their offspring were called the F1 generation. This F1 generation contained only plants with yellow peas. Mendel then crossed the F1 plants together to create the F2 generation. In the F2 generation, Mendel
So, I hope this makes sense. This was good. I was going to pull up my own chart, but they do have one here. So this is perfect. The parents, two different colors. The kids, he realized that the yellow was the dominant trait and that the green, I guess, is the recessive trait, meaning it doesn't express itself as frequently as the yellow does. And this is proven by going to the next generation, meaning the parents made the siblings. The siblings crossed and made the F2 to stabilize the genetic, not stabilize, to further show the expression of the genetics of the P1. That's all you're really doing. You going down further to find out what is actually in the genetic code. Once you find out what's in genetic code, you can kind of steer into where you want to go, get the most out of what you're trying to get from the specific traits that you're working with. So say the parents are high res, the yellow is high resin, the green is, color. It shows that the high res, the yellow, is the dominant trait because it's expressing itself through multiple generations. So that trait seems to be dominant and the one that's passing down. By the time you get to F3, F4, that's just going to be what it's going to be. It's already locked in at that point. So then you can begin to cross it with something new. And you would expect that yellow would be dominant in whatever you crossed it with. So that's kind of the scientific take notes, know the genetics of what you're working with that all those articles were talking about.
Observed that about 3/4 of the plants in the generation had yellow peas and 1/4 of the plants in the generation had green peas. The reappearance of green peas in this F2 generation told Gregor Mendel 3 things. First,
I think that it's going to be recessive. I think it's the first thing that it told him. So it's going to be not just the trait, but that one parent, I guess you sum up the P1s to just what the traits are. But it shows that the green one is not passing. So yellow's dominant, the green is recessive, and that it seems that it was a success. I'm going to say it was a successful cross because the yellow is showing up over the green. Let's see. I'm getting drunk now.
Each plant carries 2 copies of a gene that creates pea color. He knew this because these F2 green pea plants must have gotten their traits from their parents, all of which have yellow peas. So the F1 generation must have a hidden copy of the green genetics in them somewhere to pass on to the green peas in the F2 generation.
Okay. So that is something, and the reason that you keep the parents, because if he wasn't getting the results he want, I would say start from scratch, but he does have the ability to take that F2, go back to one of the parents, and start working again. I guess in essence making a new F1 instead of an F3, but it allows you to steer what traits you're going for. This is kind of the art of breeding they're talking about. You have to grow it out to see what you're getting in order to know what your next step is.
Because all the F1 plants have yellow peas, Mendel also inferred that there are two alleles for pea color, a dominant allele that encodes yellow peas and a recessive allele that encodes green peas. When a plant
Okay, the allele is what that genetic code they were talking about, the codominant traits. If you have a certain set, it works well with another certain set of genetic.
contains at least one yellow allele, the plant will make yellow peas. And when a plant has two copies of the recessive green allele, then it will have green peas. So these F1 plants are all hybrids, having received a yellow allele from this parent and a green allele from this parent. Finally, given the 3-to-1 ratio of yellow to green offspring in this F2 generation, Mendel inferred that each parent passes on just one of their two alleles to their offspring, but which of these two alleles the parent passes on is random.
And that is very true. I mean, if you think about families, I know that we don't see this as much, but siblings, even though they do have the same P1, the same parents, not every sibling is an identical twin. And exactly identical twins is extremely rare. You're more likely to get, I forget what the term is, but they're twins because they're coming at the same birth, but they're like boy and girl even. Not something twin. It starts with the S. But just because you do have the genetic code doesn't mean you're going to get an exact replica. You're trying to get as close to the replica as possible. Hence, you look just like your daddy. You look just like your mom. Okay, I see you're coming from that genetic code, but your phenotypical expression, even if it's not your genetic representation, it looks different. Your environment is making you different from your parents. And I guess what? You can get boys, girls, blondes, brunettes. That's all different phenotypical expressions, how the mom was going through the birth. The pregnancy.
So in the pea generation, this yellow pea plant is homozygous for the dominant yellow allele, and this green pea plant is homozygous for the recessive green allele. The F1 generation is made up solely of plants that are heterozygous, carrying a yellow allele from this parent and a green allele from this parent. All of these plants have yellow peas because the yellow allele is dominant to the green recessive allele.
Okay, I think I got that right. I missed the other two, but yeah, I didn't get the allele part coming. But he did figure out what was recessive and what was dominant. And he also realized the genetic potential of the original parents was in the later generations. I forgot what they fucking call that, but like, hey, you don't look like your parents, but you look like your grandparent or your great grandparent. Look at this picture of them. That's kind of this going back in time machine of genetics and trying to pick out these traits. have a cousin who on their mother's side, I think their great-grandparents had red hair. She has 4 siblings. All of them had the same color hair as me. But all of her kids have red hair. She has 3. One of them is even called Dirty Reds. He has so much red hair. So that recessive trait popped up, I guess 4 generations later. But that's what's in the genetic code.
Then, when you cross 2 F1 plants, you see that most of the offspring have yellow peas, but some do have green peas. This can be explained when you look at a Punnett square of the cross between 2 F1 plants.
Okay, the Punnett square, this is very important. These are the 2 parent probabilities of what you're going to get out of your plant, which shows dominant and recessive traits. Now I got to go whiz again, then I'm going to leave you with this and come right back. Let's get out of here. What am I recording?
This is a classic monohybrid cross. You can see from the Punnett square that you can expect to see 3 times as many yellow pea plants as you see green pea plants in the F2 generation. So as you can see, from analyzing these 3 generations of pea plants, Mendel was able to determine some key aspects of inheritance. Pretty amazing stuff. Those are the basics of Gregor Mendel's classic crosses. If you'd like to dig deeper into the monohybrid cross, see my video on monohybrid crosses. In this video, we'll walk through the P, F1, and F2 crosses that taught Gregor Mendel the basics of trait inheritance. In the 1800s, Gregor Mendel worked with pea plants to determine how traits are passed from one generation to another. One trait he focused on was pea color. He noticed that there were 2 variations in pea color, plants that produced green peas and plants that produced yellow peas.
Sorry about that. Oh, but this is perfect. I'm going to full cam. Like I meant to do that. The best stream ever. So I hope that helped to make sense on anything. Yeah, I hope that helped make sense on anything. It was about Gregor Mendel, the father of breeding. Him establishing the P1, the F1, the F2, recessive, dominant, genotype, phenotype. And that was in the 1800s. So, I mean, that's extremely impressive. I don't know if we've really advanced past. Actually, we haven't really advanced that. I'll try and remember to leave the links this time. There's also a podcast I listened to that explained Gregor Mendel very well. Like at the beginning of his life, it was from Future Cannabis Project. And that's what even got me into this. That explained his life, that he actually, at the time that he discovered all of this was ignored. And it didn't come back up until about 30 years later. I believe like the 1890s, Darwin's scholars and Mendel scholars started to battle. People coming up with this theory of breeding versus mathematician breeding theories of how plants will pass and express themselves. This isn't mathematically scientific, but as far as probabilities of plant breeding and encountering variables, this is extremely useful. So at the same time in the 1890s, I believe 3 different people came up with the same Mendel conclusion of genotype, phenotype, F1, F2, asked people about it and went back to find out that Mendel like did this 30 years ago, and that's when his work became into prominence. But at that point, how did the show put it? He hung up his cleats and became a monk like a couple years after people ignored his experiment. But that's very fascinating because we really have advanced off of that since then. What's it, Monticello, the people that make basically all the grain for everybody, and they can sue your ass if they use any of their pollen to breed. They use still Mendel's theory. They basically sell most agriculturalists F10s to F14s when it's corn, tomatoes, things like that. So now I gotta rehook up the video because I got another part for you. That was just trying to explain the father of everything, but we got more. And this is all available on YouTube. I try to make it easy for y'all. The best search engine. DaBacco University. Here we go. DaBacco University. Are we there? Okay, so.
Welcome to this DaBacco University. Looking at general.
Oh, he plugs himself. Yeah, I definitely say that. Let me turn the volume down a tad. Let me get out your face. Get into the corner real quick. And there we go. Much better. Turn it just too hard. Yeah, my head can't take that. I'm sorry. Alright, I'm with you.
Cannabis breeding overview. Just kind of looking at a general overview of something that can be quite complex. So hopefully.
Now, I love this guy. I would definitely say that. But honestly, I guess to be nice, I would expect this type of information to be coming from a person who looks like this.
This will provide a little description and allow you to develop some basic understanding.
Empty.
So first off, the basic overview, I should say, of heredity. Other videos are provided on this channel to better understand the basic breeding process, because I said it can get quite confusing.
Okay. So that sucking in air is annoying. Here we go. Just for y'all. Oh. Our other beer, I almost forgot. Hammer throw. Just imagine when you fill it up. That's like 4,000 beers. Maybe not. But the reason I picked this guy, there's a few YouTubers I try and highlight. And either I like their style or they are very knowledgeable. But what is key about him is he explains a bunch of knowledge in a short amount of time. That's what makes him so vicious. The DaBacco University. He's got a lot of videos on YouTube. He's got a lot of views, so I'm sure you might have seen him or you won't miss him. I'll leave a link.
We can look at Punnett squares and Mendelian genetics. We'll touch on that just a little bit here. Realize this is a very complex topic, and I just hope to give you that basic overview here on this video.
Of course.
So first off, we have to just understand the basics of, for example, genes. And there's dominant and there's recessive genes. So understanding the difference between those two is important. Individuals receive one version of a gene called an allele from each parent. If the alleles are different, the dominant allele will be expressed while the
Okay. So he's just substituting allele for trait. And I can understand that because it's not necessarily a trait, sometimes like one singular thing. It is a group setting, such as the entourage effect on us, a bunch of terpenes interacting with our body. It's a bunch of different genetic codes interacting with other genetic codes, getting you this. So they just call it an allele. Okay, I was a little mistaken about that. I figured it was a series of genetic codes put together, like a chain that mixes with another chain. But him signifying that really helped because I'm drunk right now. Wow.
Effect of the other allele called recessive is masked. So a gene will be expressed and can mask or override the effect of a different variant of the same gene in the other copy of the chromosome. Recessive genes, an individual must inherit 2 copies of the mutated allele in order to express that trait.
Okay. So that makes a lot of sense. Let me turn this down a little bit. That was busting it. But he's saying 2. I was figuring in my head it was anywhere from like 3 to 6. So let's see. If this was how they would match up, let's see. So that, let's say that. So these 2 alleles mixing up. Oh shoot. Okay. So hold on. Wait a minute. No. Yeah. These 2 alleles mixing up. Basically, it's a perfect combination to get you the result you're looking for. That's the like licorice. That's the pink hairs. That's the pineapple expression, the chocolate expression. It's those specific alleles locking together 3 to 4 specific traits that work well together. So let's get you.
Now, keep in mind, this does not mean that genes are bad or negative recessive genes. Some are, but not necessarily all of them. Sometimes recessive and dominant genes here have really no major effect.
Honestly, I think that's not true. On one of the podcasts I was listening to, I want to say it was Breeder Syndicate. They have a series on breeding cannabis, growing your own strains, and a lot of cannabis is that we are trying to unlock these recessive traits. The rare and unique taste and flavors and effect are mostly recessive. So we're trying to take that recessive and make it dominant. Like a strawberry flavor, a pineapple flavor, a guava, an apricot. Those are rare flavors. So you're trying to take that recessive rareness and make that more dominant.
Where we can see the example here of having a attached earlobe versus kind of a distinctive earlobe here. This is an example of recessive trait and a dominant trait. Doesn't mean that the recessive can hear any worse than the dominant. This is an example of a phenotype, or what it would physically look like compared to the genotype, the genetics here, and we can trace that through different lineages.
So he said that I didn't think that, yeah, one's not better than the other. Because you don't have apricot doesn't make it bad weed. But if that's what you're going for, that's how you get these unique vanilla, chocolate, creamy, hashy, different tastes. Those are recessive.
Now you get into things called sometimes true breeding. And true breeding is a kind of breeding where the parents would produce offspring that would carry the same phenotype. Again, phenotype is how the organism looks. This means that the parents are homozygous for every trait. In this example, we're breeding purple with purple, getting purple, breeding purple with purple, getting purple, so on and so forth. They're true breeding. They come true from type. We see the same thing here with the white flowers as well, where we're noticing that the self-pollination, we're getting more and more of the same. These would be examples of true breeding lines. Keep in mind there's 2 sex chromosomes. We identify a female as having 2 X's and a male as having an X.
I'm
and
trying
Y.
to get out your
This
face.
is how we get the female flowers, as we can see here, and then the
There we
male
go.
flowers we can see closer to me here. This is again
I
boy
hope
and
that's
girl,
better.
same example. Now the Y is drawn smaller, at least in humans, because the Y chromosome does contain less genes than the larger X. Now, a simplified Punnett square, and this may take you back to kind of biology class, where you're looking at crossing.
Now, see, he goes into the Punnett square. This is something you really can't avoid. Each square vertically is the one parent. So these are the probabilities that you get. The 50-50 of both traits, one representing the mother and one representing the father, however you want to explain it, just to go to traditional breeding, making it easy. But if it was even female, taking a clone, 2 clone of a parent plant, breeding them, you're going to get this probability is going to come up. Is it going to express it's going to express all of the genetics that are inside the parent. Phenotypically, when you grow it, you're going to get to see what it has, all the potential.
2 genotypes together, and this Punnett square is just a quick way to organize things. And it's a great way to kind of quickly go through things. We see the big G would be dominant over the little g. We have a heterozygous, this is called, where we have a dominant and recessive allele. With a dominant recessive allele, we breed them together. 25%, or 1 out of 4, would be the homozygous dominant. 50% would be heterozygotes. We have 1 dominant and 1 recessive allele. And then 25% would be homozygous recessive. Now, phenotypically, or how they would look, 75% of these would be green if the capital G represents green color, and 25% would be yellow because they need to have both inherit both of those genes there. Again, just a simplified, quick run through of Punnett squares. You might hear things called like the F1 and the F2 generation because some traits may skip a generation. So here we have the parental generation, the F1 and the F2. In this example, the parental generation, we have a tall plant and a short plant. You can see their genotype here. When these are bred together, all the resulting offspring are tall, but they're all heterozygotes, meaning they all contain that masked or that hidden recessive allele. However, when we breed these together, we see those short varieties come back again. They skipped that generation there. So it's important to keep that breeding lineage going through.
So that's what I was basically talking about, being able to skip into different genetic sessions. So keeping the seeds, keeping the parents are very important.
And we can see that here again with the kind of cannabis strain, 2 cannabis strains, breeding those together, where we're tracking everything. Use a female from these seeds, make a back cross. Talk a second about back crossing, but keeping a lineage and kind of a family tree is important for tracking traits. Now that back crossing that I briefly mentioned previously, back crossing is crossing of a hybrid with one of its parents or an individually genetically similar to its parent in order to achieve offspring with a generic identity which is closer to that of the parent. It's kind of like going backwards, literally within the cross. So here we have our parental strain, we have our F1 strain. Well, we have that back cross going.
Literally going backwards. So that's key. When you see a BX4, which people say isn't really real or whatever, but they're trying to say how many times they went back to the parent to lock in the specific trait. I think of Cherry Pie BX4. I've seen that. And that basically means through 4 different successions of growth, from veg to flower, he cloned. He just kept going back to the parent, back to the parent, back to the parent, to lock in that cherry, that specific flavor he was looking for. Nothing stopping you from going whatever specific trait you're looking for. You're trying to take the rare, build it up through back crossing or forward generations, taking the parents and the F1s of what they produce, going F2 if you're finding success in the F1s, and if you're not, going back to the parent to try and lock in what you are looking for.
Back through and reverting it to one of the parental there. And that's helping limit the kind of variability that you get. So here we have two parents. This is kind of a good way to kind of visually consider it. Parents A and parents R, breed them together, we get offspring B. We're going to breed offspring B with that recurrent parent, parent R again. B with R, we get C. And then C, we breed that with R again. And we're kind of back crossing the sense we're going back to those genetics. We're constantly bringing those back into the line to try to create some genetic variability, but also kind of keep an idea of some of those traits recurring back in the population. Now, Gregor Mendel was a monk that studied pea plants.
Okay, my bad. I said bishop. It was a monk. I knew it was something important and he was religious. But he was German, like the 1850s when he came up with this. That's the first video that we watched was referring to his experiment, and here it comes up again. This is at least a 2020, probably 2023 video, so we haven't really gone that far in the 100-some years since Mendel.
These are kind of an example of kind of dihybrid crosses are called. So again, this is a simplified example. The phenotypes are provided in the simplified example. It's kind of a great visual that we can see. Tall plants and green plants looking here. Many traits do not follow Mendelian inheritance, so it's important to keep that in mind. So this helps show the variability that can occur from a basic cross involving only two traits. Now when I say they...
All right, I'm sorry. You can watch the video later, but is it just me, or is his voice, like his cadence, putting you to sleep and almost like knocking you out?
Don't inherit all Mendelian. So for example, here we either have green or we have kind of that purple color. Well, there's a chance we could get green with purple spots or purple with green spots. So there is a lot of complexities. And while you look at this and go, well, this is pretty complex, this is actually a simplified version of having either a trait being expressed or not expressed, following those Mendelian inheritance. They're either tall plants or they're short plants. They're either green or they're purple. So this is why keeping a track record of your breeding projects, having a good idea going in and reviewing this video as well as other videos that are on this channel.
Okay, he just plugging himself. I can tell by the little meat that it's about to be old. So let me get out of this shit. Let me do this real quick. My bad. Oh, that actually helped. Since my eyes was all puffed up and I can't see. Okay, cool. So, as I was saying out there, Gregor Mendel comes up again from something that happened in the 1850s. What was you doing in 1853, dude? His pea experiment lives on. So we just watched two videos still covering something from the 1800s. Squeezing that juice. But basically saying keeping track records, paying close attention to the results that you're getting out of the plant, growth rate, vigor, color. And also environment. He kept strategic records of the temperature, the barometric pressure. Once he realized that, hold on, the parent's genetics is in the F2, I gotta make sure I know all of it. And his experiment, his theory, seemed to actually kind of hold true. So let me get you on to our next video. Trust me, I got you covered. We listen to all them dummies talk. I shouldn't say dummy. That was mean. Do I have one more video? I think I feel like the F3. We went through P1s, F1. Oh yeah, I really can't. Almost got to get up in your face. Okay, I know it ain't this. No, okay. Let's say F3. No, not that. Oh, my eyes is hurting. Oh, I still had. Here we go. It was blank, nothing to go off of. So that's why I was sorry to find. Okay, hold on. And this seemed to be giving me problems.
Hello and welcome.
Okay, there we go. I heard Russian. At this about probability, this is going to be right. So we're about to watch a video about the F3 generation. Now you start with P1, which is the parent. Purple Haze and Bubba Kush. When you cross them together, you get the F1 generation. It's going to be 25% Bubba Kush, 25% Purple Haze, and 50% mixture. Depending on what you want, you got to sift through that and figure it out. Once you find a few that you like, ones that you like from that F1 generation, you cross them together. That makes the F2 generation. Now you do this to kind of see how over time the plants are going to respond. Once you start to see it's going in the direction you want it to go, you can go into the F3 generation. Back crossing comes up if you don't find what you're looking for and can't get to the F3 generation. You need to go back to the F1 or either start over from the parent, the original crop, and try and sift. But the F3 is basically the point of no return, where you're going to lock in the traits that you are looking for. What it is what it is at this point. F4 is just to make sure that what you think is happening is really happening. So F1 is the mix. F2 is to select from that mix. F3, this is it. Point of no return. Let's go. Oh, that was my cool DJ Rattler. So let's get into... Did that work? Oh, that didn't. I can't just switch it up. Oh, already ready. All right. Here we go. Yeah, I'll stay here. Screw it. Best streamer of all time.
Welcome to Nikolai's Genetics Lessons, and today's problem I got from one of my subscribers, and here's the problem. In a cross, the plants of F2 generation segregate 3/4 red and 1/4 white. If red plant of the F2 generation has self-fertilized, what is the probability that both phenotypes will be observed?
Whoa, that was tough. But yeah, I got a whiz. I can't think right now. So give me a second, and I'll be right back. Alright. Oh, Nikki, I need you now. Alright, my bad. I did not expect that. My wife hit me up. Let's get into it.
And its progeny. As you see, the red pigment is dominant and white pigment is recessive. So let's start with the parental generation. So we have one plant of the genotype capital A, and we cross with another plant which is small a, small a. So we call this parent 1 and parent 2. And phenotype of this plant is red, and phenotype of this plant is white.
Okay. So one's recessive, one's dominant. If you can remember the Punnett square, this is the top portion of it, and they're going to fill in the bottom two portion.
And next generation is going to be F1 generation. So what we are going to see in F1 generation, that all plants are going to be heterozygous. So from parent 1 they can get only dominant allele, and from parent 2 they only can get recessive allele. And phenotype is going to be
Heterozygous is what same species you're going to see a variation versus, say, a land race which will be more homozygous. You're only going to see specific things. There's going to be a lot of different expression in this.
red of all these plants. And when we self-pollinate them, we are going to get next generation, which we call F2 generation. So in F2 generation, we are going to get following genotypes and phenotypes. So one parent and another parent, both of them are of the same genotype. So let's build simple Punnett square, and let's take a look what we are going to see in F2 generation. What
it's a second Punnett square. That's
genotypes.
already one up top.
Capital A here, capital A, small a here, capital A, small a here, and small a here. In F2 generation, we are going to see three.
And this is probabilities of what you're going to see. How you saying one is red, one is white. This is the probabilities how much you're going to see the white, which was going to be the rare, the recessive.
Genotypes 1, 2, and 3 genotypes, but 2 phenotypes. So this is going to be one phenotype here. So these 2 genotypes would produce plants with red pigment, and this one is going to produce this genotype flowers with white pigment. As you see, the ratio of phenotypes is going to be 3 to 1. And now let's return to our problem. If a red plant from the F2 generation has self-fertilized, what is the probability that both phenotypes will be observed in its progeny? So that means that we have to exclude this variant here. We now only work with this sample space. So we also exclude this ratio here. And now we are working with frequencies or ratios not of the phenotypes but genotypes, because we work with one phenotype. And ratio of the genotypes is going to be 1, which is homozygous dominant, to 2, which is going to be heterozygous. So ratio is 1 to 2, 1 to 2, or we can say 1/3 and